Good morning, everyone. Now, up to this point, we talked about clinical research. Now, what I'm going to talk to you about has to do with neuroactive steroids and their role in ECT. Now, our presentation plan would start with a definition of steroids, neuroactive steroids, history, biosynthesis, and the receptors on which they have an effect, what kinds of clinical studies there are out there, of course with a major depression lens, and finally we will have a look at the relationship between NAS and ECT. Now we know that steroids are synthesized from cholesterol and adrenal glands, gonads and others synthesize these. And they have important roles in the maturization of the brain, for instance. Steroid hormones bind to intercellular receptors, have an effect on gene expression. It's called genomic effect, and in order to have these genomic effects we need protein synthesis, and it might require minutes, sometimes hours. But certain steroids do not have genomic effect, but they have non-genomic effect, as we call them, and they have an impact on the receptors that are outside the cells and modify certain neuronal excitability, and this can happen in seconds or even milliseconds, so very, very short. And these steroids are called neuroactive steroids. They don't have genomic effects, they rather bind to receptors that are on the surface of the cells, so they seem to be slightly different than steroids. Now when we look at the history of steroids, Cushing is the first name that comes up. He focused on emotions and endocrine secretion, and he comes up with a pathology specific to himself. But is it emotions that change secretion, or is it the other way around? In fact, both seem to be correct. In 1932, he made an observation. He focused on adrenal hyperactivity patients, lack of sleep, concentration difficulty, and depression periods. He found out that there are periods of anger in these patients. And then Beaulieu had certain examples of research in this field. Now, steroids have genomic effects and on the nucleosis, and he works with adult rats using DHEAS, and so that the concentration is 20 times higher in the brain. So maybe is there additional synthesis in the brain? Now, again, in rats, in the brains of rats, neuroactive steroids have been shown at high levels. And as a result of his evidence, he turned to steroids that showed their effects on the brain and can be synthesized spontaneously in the CNS from cholesterol or the peripheral steroid precursors that can easily cross the blood-brain barrier as neurosteroids or neuroactive steroids. Now, as I was saying, fetoplacental unity, adrenal glands, the ovaries can help with the secretion of these, but is it maybe also secreted through the CNS? So that's what he found. So how is it secreted? Well, from cholesterol through a series of enzymatic reactions, and it's a pathway that we are familiar with: glucocorticoids, mineralocorticoids, and sex hormones. These are what they are transformed into in the brain. Where does this happen? In the peripheral peripheral nervous system neurons, and in the brain oligodendrocytes, 1 cell, and type 1 astrocytes, and their mitochondria rather. At the beginning of neurosteroid synthesis, cholesterol crosses into the mitochondria via peripheral type benzodiazepine receptors that locate on the outer membrane of the mitochondrion. And then the first step of synthesis, cholesterol's conversion into pregnenolone by cytochrome P450 SCC. Now, if we are to demonstrate what actually takes place on this chart, it enters into mitochondrion and turns into pregnenolone. And then I'll skip the names of the enzymes, but with the use of a number of enzymes, it turns into progesterone, for instance, deoxycorticosterone, and 3-alpha-5-alpha tetroprogesterone, or allopregnanolone, and others. All of these are active neuroactive steroids, so they correspond to something in the brain. This is another chart we can have a look at. Cholesterol enters into mitochondrion, turns into pregnenolone, and once outside, it turns into certain neuroactive steroids with the help of a number of enzymes. Now, it's not, of course, only the ones I've listed. For instance, we also have aldosterone, corticosterone, and cortisol, which follow the same pathway. Estradiol, testosterone. All of these substances are actually neuroactive steroids in that they have an effect on the brain. And there are lots of studies focusing on lots of receptors to see the mechanism of action. I'm going to simply talk about GABA because it is very detailed in terms of its effects, but the most important is GABA A. We have sigma, glycine, serotonin, nicotinic, and muscarinic receptors, NMDA, kainate receptors. So there are lots of studies showing that this actually has an impact on all of these receptors. GABA A receptors constitutes the most important group for us. It was for the first time in 1986 that they were found to be in interaction with neuroactive steroids, estradiol and other known neuroactive steroids, of course. And here we can talk about rapid synaptic activation in the central nervous system, mostly occurring due to the activation of GABA receptors. And it's also been demonstrated that neuroactive steroids were bound to a site on GABA A receptors, distinct from where GABA, benzodiazepines, and barbiturates bind, and had a distinctive binding site. And in this way, it has anxiolytic, analgesic, anticonvulsant, sedative, and anesthetic effects. It also protects neurons. It also helps with neuron augmentation and increases synapse generation. This is simply a chart showing the genomic effect. As you know, it enters into the cell, and in the nucleus of the cell, it works on the DNA, on microRNA, mRNA, and then we have protein synthesis, and this can take days or even hours or even days. Neuroactive steroids, however, are very quick. They can act in seconds or minutes because they act on the GABA A receptor on the cell membrane, and their effect is thus instantaneous. This is a similar pathway, and once this happens, we get pregnenolone, let's say, from cholesterol, and it then binds on the receptors on the cell membrane and has an effect in that way. Now, lots of substances that we know as neuroactive steroids can have negative or positive interaction with a number of receptors. This is a summary. Progesterone, for instance, has positive effect on kinates, but negative effect on glycine, 5-HT3, nicotinic acetylcholine, and oxytocin. And you see the other substances. Mostly, as you clearly see, they have their effects through GABA. Let's have a look at the clinical side. It applies in mood disorders especially, but also in anxiety disorders, psychotic disorders, dementia and memory disorders, alcohol addiction and withdrawal, eating disorders, and pain. There are lots of studies in these areas considering each of the neuroactive steroids. Today I'm going to talk about mood disorders. In major depression, for instance, alterations of NAS levels in depression are reported by several studies. It is demonstrated that plasma and CSF, allopregnanolone and pregnenolone levels were significantly lower in patients with depression, and those levels reached a normal range after treatment with fluoxetine and fluvoxamine and the non-SSRI antidepressants. This has been demonstrated in studies. Similarly, in rat models of depression, it is observed that antidepressant effect had occurred following allopregnanolone medication. In other studies on depression, DHEAS is a significant substance. Certain studies have conflicting conclusions, but they say that this substance tends to increase, its levels tend to increase in depression. Again, in patients with DHEA administration, significant decrease in Hamilton Depression Rating Scale, or HDRS score, was reported. In studies that assess testosterone, it has been demonstrated that testosterone levels were lower in depressive male patients, and depression symptoms had improved following testosterone treatment. In another study that was conducted on hypogonadal men, it was reported that testosterone replacement therapy had decreased fatigue and jitteriness and improved the emotional status and rehabilitated the depression. Despite all this research, the hypothesis was reviewed again. The hypothesis for depression, that is. Although clinical improvement was observed in patients in which depression treatment was managed by transcranial magnetic stimulation, partial sleep deprivation, and electroconvulsive therapy, alterations in plasma neuroactive steroid levels were not, however, observed despite the recovery or improvement as a result of these treatments. Dubrovsky is a leading name in the field, and he suggests that the clinical improvement, despite the absence of alterations in plasma neuroactive steroid levels, might indicate to a paracrine effect, so they hormone secrete substance that can have an immediate effect on the surroundings, and that might be a mechanism to explain this situation. And there are lots of studies in the field. So what's the relationship between ECT and NAS? ECT is maybe the most effective method that we have in the treatment of depression, but it's still unclear how antidepressant effect of ECT actually takes place. And may neuroactive steroids be one of the factors that play a role in forming the antidepressant effects of ECT? There are a number of studies since the year 2000 about this field. Not so many studies, but it can be an interesting field for studies. This is one of the first studies in the field. And it has 31 medication-resistant major depression patients. They received unilateral ECT for 4 weeks, and pre- and post-treatment substance levels were controlled for the following substances, and they concluded that there was no change in the levels of these substances. Another study with 17 patients compared to 25 healthy controls, and the common feature of these patients is that they are depressive. 2 of them have psychotic major depression, 10 have schizophrenia plus comorbid depression, 5 have schizoaffective disorder. So they checked pre-ECT and after 6th session of ECT. Cortisol, DHEA, and DHEAS levels were checked, both at the basal level and after the ECT. All levels were found to be higher. And the increase in DHEAS levels has been shown to be associated with being resistant to ECT. This seems to be an important piece of information. So the DHEAS levels at the basal level have been associated with resistance to ECT. They tried the same thing on animal models this time around, and they asked the question to see if the levels of the DHEAS are associated with resistance to ECT. They used rats. The rats received DHEA injections for 13 days, and they received electroconvulsive shock, which is the analog for ECT, and the result is that they end up with high basal DHEAS levels, or rather these levels are actually associated with resistance to ECT. There's a study from Turkey from 2008. They had 25 patients diagnosed as MDD and who responded to ECT. These were compared with 37 healthy individuals. Cortisol, DHEAS, 17-OHP, and testosterone levels were measured on 2 days before the first ECT administration, 10 minutes after the procedure, and 3 days after the last ECT administration. And again, basal plasma DHEAS levels were higher, according to the study, in patients with depression controlled compared to the controls, and the same occurs time and again in other studies. 17-OHP and testosterone levels were lower. This also seems to be in harmony with what we already know about the case. Cortisol levels were similar, and basal DHEAS levels became more markedly increased after the administration of ECT. We have our own study as well about this point, and we had 19 treatment-resistant NDD patients that had not been using antidepressant drugs for the last 1 week, major depressant patients. Plasma progesterone, testosterone, pregnenolone, DHEA and DHEAS and estradiol levels were assessed, and all levels were normal. After the ECT, however, only in men, DHEAS levels were significantly increased compared to the measurement prior to the ECT. So to give you a summary, NAS or neuroactive steroids actually engage the receptors on the cell surface, and they have non-genomic effects very rapidly, and the onset happens within seconds. So this is a different feature that they have than steroids. They're related with affection disorders, anxiety disorders, psychotic disorders, dementia and memory disorders, alcohol dependence and abstinence, eating disorders, and pain. And there are studies in each of these fields. Similarly, alterations in NAS levels may be one of the factors that play a role in antidepressant effects of ECT. Similarly, it's suggested that elevation in basal DHEAS levels is related with the resistance of ECT, or it has been claimed according to different studies. Of course, we need further research, further and larger samples, because the studies I've shown you, they had a maximum of maybe 30 cases. So this seems to be a promising field of research. I'd like to thank you for your attention.